Hello everybody, you are welcome. Today we will solve together paper 3, paper 33, May, June 18. Before you proceed, read carefully through the whole of question 1 and question 2. Plan the use of the 2 hours to make sure that you finish all the work that you would like to do. If you have enough time, think about how you can improve the confidence in your results. For example, by obtaining and recording one or more additional measurements. You will gain marks for recording your results according to the instructions. Let's start. Number 1. Fruit growers need to know the concentration of sugars, such as sucrose, in the fruit so that it can be picked at the best time. As the fruit matures, the concentration of sucrose increases. The concentration of sucrose can be estimated in fruit extract by carrying out the non-reducing sugar test. Known concentration of sucrose are tested, and the result for the unknown concentration of sucrose in the fruit extract is compared to them. You will need to prepare known concentration of sucrose solution using simple or proportional dilution. Carry out the non-reducing sugar test on these concentrations. Carry out the non-reducing sugar test on the unknown concentrations of sucrose solution in fruit extract U. Finally, estimate the concentration of sucrose in fruit extract U. Okay? You are provided with this material shown in table 1.1. S5, W, U, H, A, and Benedict's reagent with their contents. Here, look, sucrose S5 is sucrose with concentration 5%, okay, about 60 cubic centimeter. In small beaker containing about 60 cubic centimeter with non-hazards. W, distilled water, also with non-hazards, with volume equal 100 cubic centimeter. U, unknown concentration of sucrose solution, about 20 cubic centimeter. H, dilute hydrochloric acid, it's irritant, okay, irritant because it is strong acid, okay, with volume about 50 cubic centimeter. A, 10 gram of sodium hydrogen carbonate powder in petri dish with non-hazards. Sodium hydrogen carbonate powder also we use it in baking powder. When baking cake, for example, a packet of baking powder containing sodium hydrogen carbonate powder. So it's without hazard. Finally, Benedict solution. As you know, Benedict solution is dark blue in color. Okay, dark blue in color. It is irritant also and harmful in the volume about 20 cubic centimeters in your exam. Okay, it is recommended that you wear suitable eye protection, eye goggles. If H, A, or Benedicts come into contact with your skin, wash off immediately under cold water. It's okay. Read step 1 to step 19 before proceeding. Number 1, set up water bath and heat to boiling ready for step 7 and step 13. Now, how can you prepare your water bath during exam? Look, we use Bunsen burner, tripod, okay, and metallic mesh or gauze mesh, beaker containing enough amount of tap water and thermometer. Okay, please, during preparing your water bath, fill the beaker, the large beaker. You are provided with large beaker for water bath. Fill it with enough amount and make sure that the test tube placed inside not floating. Okay, and all the contents of reagents inside test tube must be immersed, must be covered by water inside the water bath. Okay. Now, another method for preparing water bath by using hot plate, electric hot plate like this one. It's okay. Now, 4. you will need to make simple proportional dilutions of the sucrose solution S5 and prepare the concentration of sucrose solution. Prepare 4% sucrose solution or sucrose concentration to be labeled S4. 3% sucrose concentration to be labeled S3. 2% sucrose concentration to be labeled S2. You will need to prepare 10 cubic centimeter of each concentration. So the enough amount needed to carry out your steps in your exam is 10 cubic centimeter from each reagent. Only 10. It's okay? How? Now, with this skill, this important skill, how to prepare simple or proportional dilution. Okay? Letter A, single I. Complete table 1.2 to show how you will prepare these concentrations. For S5, label here S5, S4, S3, S2. You have to label small beakers provided at your exam or at your bench by permanent marker S5, S4, S3, S2. Okay, here volume of S5 per cubic centimeter, the unit in headings, not in cells of the table, equal 10 cubic centimeter of S, and the volume of distilled water W 0 added. So that's concentrated solution that you are given at your exam. Final percentage concentration of sucrose in this beaker equal 5%. Okay, 5%. Now how to prepare S4 with 4%, S3 with 3%, S2 with 2%. How? We use certain formula. Stock to required. What's the formula? C1V1 equals C2V2. C1V1 for stock, C2V2 for required volume and required concentration. Now C1 compensate. C1 equal 5%. This is the stock given at your exam, 5%. Times V1, the volume needed to be taken from stock solution, I mean 5%, okay, equal 4%. The required volume, the required concentration here 4%, and the required total volume needed to carry out your exam 10 cubic centimeter. So V1 equal 4% times 10 cubic centimeter divide 5%. Okay, so the result equal 8 cubic centimeter. 8 cubic centimeter added here with 1 decimal point, like the figure 10.0. Exactly imitate the examiner, please. Here 8.0. Okay, so what is the remaining volume to reach 10, total 10? We need to add 2 cubic centimeter of water. Okay, this is the first dilution, equal 4%. Now how to prepare S3 with 3%? Please compensate 3% where? Here, instead of 4. Instead of 4. So instead of this figure, we replace this by 3. 3 times 10 equal 30. Divide 5 equal 6. So the answer here in the table 6. What the remaining volume needed to complete your desired volume? 4. So 4 plus 6 equal 10 also. Now for the third one, compensate instead of 4 or 3 instead by 2. 2 times 10 equal 20. 20 over or divide 5 equal what? Equal 4. So the remaining volume taken from distilled water equal 6. Okay? Okay. Number 2, prepare the concentration of sucrose as stated in table 1.2 in the beakers provided. The sucrose concentration can be estimated by using the non-reducing sugar test. To test the presence of a non-reducing sugar, any non-reducing sugars must be hydrolyzed firstly, broken down into the reducing sugars, I mean monosaccharides, by using certain enzyme called sucrase enzyme or by using HCl then heating, dilute hydrochloric acid then heating. For example, boiling sucrose with dilute hydrochloric acid hydrolyzes sucrose into glucose and fructose, which are both reducing sugars. They are source of electrons to reduce Cu plus 2 found in Benedict solution, okay, into Cu plus 1. Cu plus 2 is blue in color. Cu plus 1 is red in color. Okay, red in color. Red plus blue makes the solution greenish than the other positive results, as we see later. The Benedict's test can then be used to show the presence of these reducing sugars. In steps, here we label 5 test tubes: S5, S4, S3, S2. Okay, and the last one for unknown sample of sucrose concentration. Here we add for each test tube 2 cubic centimeter of sample of sugar, and we add in step number 4 we add or put 2 cubic centimeter of HCl, dilute hydrochloric acid. Shake the content of each test tube. Just swing the test tube, okay, 2 times, 3 times, no problem, and placed in the beaker later. Okay, shake the test tube gently to mix the contents. Repeat step 3 to step 5 for S4, S3, S2, and U. Okay, now number 7, put all the test tubes into the boiling water bath. Boiling water bath. Set up in step 1 as we saw before. Leave them for 2 minutes. 2 minutes in water bath. Okay? 2 minutes in boiling water bath. Now after 2 minutes, remove the test tubes from the water bath and put them in test tube rack. For what? For cooling. Look. Leave the test tube to cool for further 3 minutes for cooling. You will need the boiling water bath for step 13. Okay? After 3 minutes of waiting for cooling these 5 test tubes, we need to make certain essential process called neutralization. Why neutralization? Because the contents from S1, S5, S4, S3, S2, and U are acidic due to dilute HCl. Benedict solution, okay, act and work perfectly in alkaline medium. So we have to change the medium of these 5 test tubes from acidic medium, strong acidic medium due to HCl, into neutral 7, and even maybe reach 8 or 9. Okay, now after 3 minutes, put small amount of I mean sodium hydrogen carbonate. Okay, here in this exam we neutralize HCl content, okay, acidic content of each test tube by using powder of sodium hydrogen carbonate. How come? By adding, okay, small pieces of powder, of white powder of sodium hydrogen carbonate. Just sodium hydrogen carbonate reach the content of each test tube, fizzing takes place. Why? Because this, remind yourself with chemistry, this is the acidity test. Okay, fizzing occurs due to release of carbon dioxide. Okay, so how we reach the end of neutralization? How you can you recognize the end of neutralization? Finishing or ending of fizzing. This means that no acidic medium. Now it is neutral or becomes what? Becomes alkaline solution. Okay? If we add amount of sodium hydrogen carbonate solution and the fizzing still present, add extra and successive amount till ending and no fizzing takes place, no bubbles takes place. This means we reach the desired point. Okay, now the mixture will fizz and rise up the test tube. Repeat until there is no more fizzing. This neutralizes the acid so that the Benedict solution can work. Okay, now note this hint or not. There may be some of A left, some of powder left at the bottom, in the bottom of some of the test tubes. This will not affect the results. Okay, don't fear. This does not affect the result. Then we put 3 cubic centimeter of Benedict solution into the test tube containing S5. S5 here, after neutralization, cooling, then neutralization, we add 3 cubic centimeter of Benedict solution added to S5. Some residue, some white precipitate here. Okay, this not affect the results. Shake the test tube gently to mix the contents of S5 containing sucrose solution, containing sucrose sugar, and the Benedict solution. Just mixing, okay, after shaking, put this test tube in the boiling water bath. Start timing by your stopwatch. Measures time taken to the first appearance of a color change in the test tube. If there is no color change after 120 seconds, stop timing and record as more than 120. Okay, your results. Record the result in II. Please draw your table before running your experiment. Read carefully for the first 5 or 8 minutes, okay, at the start of exam. Okay, 8 all the steps and make your table firstly and prepare yourself. Then record your answer easily in this table. Record the result in II. Remove the test tube from the water bath and put it in the test tube rack. Okay, now we have finished S5 detection, the end point of S5. How come? Look, we put S5 containing S5 and the Benedict solution in water bath and start timing. Just we put this test tube in water bath, boiling water bath with thermometer inside. Okay, you record the time taken to change the color, the first appearance of change. Look at the bluish color, the whole test tube, while at the bottom greenish area appear. This is the end point. So record this number as 20 seconds. Okay, now repeat step 11 to step 16 with each of the other concentration of sucrose solution. Repeat step 11 to 14 with the sample from U. Record the result for U in III. Look now, here a summary of steps. We put sample of sucrose solution from beakers into test tubes, 2 cubic centimeter. Then we add 2 cubic dilute HCl for each test tube. Then we put all the test tube in water bath. This acidification and hydrolysis of sugar present and disaccharide present in this test tubes. Then cooling, okay, at test tube rack. Then neutralizing, okay, till no fizzing takes place or no bubbles appears. This is the end of neutralization. Then 30 seconds for S3, 40 seconds, 45 seconds for S2, 70 seconds. What about you? Look at the appearance of greenish color as near the bottom of the test tube, because it is near the hot plate or near the Bunsen burner flame. Most probable appear firstly at the bottom. Okay, look now for you. For you, record about 38 without fraction seconds without fraction. Please, when we deal in our exam with seconds, no fractions written in your answers. Okay, whole number 4 seconds. It's okay. Now, all of these figures just examples. Okay, imagine that you carry out this experiment and obtain these results. Look now, record your results for known concentration of sucrose solution in an appropriate table. What about the table? The table, when we draw a table, please obey, okay, these instructions for headings. Headings: percentage concentration of sucrose appear. Heading: time needed or required for endpoints to change the color of Benedict from blue into other colors or greenish color, okay, per second. Time for all sucrose concentration S5, S4, S3, S2, and U must be recorded. The highest sucrose concentration S5 must have the shortest time to first color appear or color change. Okay, shortest time. Now, all times must be in whole seconds. These instructions to answer and obtain full mark with 5 marks. Okay, look at the table. This predicted table: percentage concentration of sucrose 5, 3, 2 with 1 decimal point, and U also with the second recorded by your stopwatch. Okay, all of these figures in trend. Okay, just in trend. Now look, triple I states the time taken for the appearance of the first color change for U. Complete figure 1.1 to show an estimate of the percentage concentration of sucrose solution in the sample U using the letter U. Here's the scale from 0 to 5%. Okay, this percentage concentration of sucrose solution U, where U be written on this scale. Okay, actually, okay, here how come state time taken for U. Okay, 38 seconds, for example. Okay, decide on estimated concentration from table. Then mark U on figure 1.1 scale between 0 and 5. Where precisely we put letter U. Actually, as biology instructor or supervisor, I prepared you before the exam as what? As 3.5%, and you are given as unknown. Okay, so for teacher it is known, for students, for you it is unknown, and it needed to be estimated. I already prepared this, okay, 3.5%, and label the beaker with U, with unknown sample. Okay, so guess where 3.5 lie? Here, in this area, in between 3 and 4. So U must be written within 3 and 4. Okay, in between. Now, the concentration of sucrose in U can also be estimated from graph of your results. Draw a graph using the results you recorded in AII, where here, this is the result in AII on the grid in figure 1.2. The axes have been labeled for you. Thank you. Use sharp pencil for drawing graph. How come? Look, I use these figures. 1, 2, 3, 4, 5 as percentage concentration of sucrose from 0 to 5. What about time? The largest or the biggest time here 70. So I choose on y-axis the dependent variable 20 for 10 small squares. For each small square with 2, 20 over 10 equal to 40, 60, and 80. Then plot small crosses or dots surrounded by small circle. Okay, crosses or dots with circles. Okay, Now here 5 with 20%, sorry, with 20 seconds. 4 with 30 seconds. 3 with 45 seconds. Okay, I think 3 must shift it one to the left, one small square. It's okay, no problem. 2 with 70. 2 with 70 seconds. Now we can plot our graph by connection, by your hand, or ruled, no problem. Okay, like this one, then. Okay. Here how we can also estimate from the graph. Look, we got that the time taken for at end point equal 38 seconds. Where 38? Here. Touch your curve at this point. So down to reach the concentration of equal also 3.5. Okay? 3.5. Okay. Here, use your graph and the time stated to estimate the percentage concentration of sucrose in show on your graph as we done in the last slide, equal 3.5. Okay? Use the time for to determine concentration from graph. So working on the graph like we did. Okay, correct estimated concentration of value 3.5%. Okay. Now identify one significant source of error in this investigation. Identify one significant source of error in this investigation. Here as color appear or color change is this source of error because it is subjective. It differs from one student to another. Okay. May you take or record say 20 seconds. Your classmate, okay, may record 30 or 25 or 27 or 15. This source of error because unable to detect or judging or identify first appearance of color change. Okay, this common in all experiment that in its observation there is a change in color. Okay, this source of error is common, where difficulty identifying first appearance of color change from blue to green. Okay, it differs from one to one. It is subjective because it depends on naked eye. Okay, now for VII, a student carried out the same procedure to estimate the percentage concentration of sucrose solution in different fruit extracts. The estimate for the percentage concentration of sucrose solution was found to be below 2%. You are given different fruit extracts as unknown. The estimate for percentage concentration of sucrose solution was found to be below 2%. How you recognize that is below 2%? Because the time record. Okay, time record what? More than 120 seconds. So of sure it contains little concentration, less than 2%. Okay, now apart from drawing graph suggests how the student could improve this procedure to obtain a more accurate estimate of the percentage concentration of sucrose solution in this fruit extract. How the student could improve? Of sure, as the estimate of this unknown concentration of sucrose below 2%, so no need to prepare large concentration like 5, like 4, like 3, even 2. So we need smaller concentration, okay, than 2. How many concentration? Many. At least 5 concentration below 2 or at highest concentration 2. Look, increase the number of sucrose concentration below 2%. Like what? Like 0.4, 0.8, 1.2, 1.6, and 2. At least 5 concentrations. Okay? Any valid points. Use smaller intervals between low concentrations. This increase accuracy in your results. Okay, accuracy. As we use smaller intervals, we can detect or estimate the unknown sample irrespective of your exam. Okay, question. We can detect accuracy. We can increase accuracy. By what? By using smaller intervals to detect the color change. Okay, precisely. Okay. I would like to discuss something about accuracy. Accuracy means the result of the test, the observations of the test, near or close to the actual. As you are close to the actual, you are more accurate. Okay, more accurate. Now, for letter B. Maturing fruits contain many types of sugar. The proportion of these different sugars vary according to the type of fruit. Of sure. Apple different from orange, different from grapefruits, grapes. Okay? The scientists carried out some tests to determine the mass of each type of sugar found in one type of fruit, only one type. The total mass taken by the scientists of the fruit was 55 grams, only 55 grams. The results are shown in table 1.3. Type of sugar in the fruit, different types. May be sucrose, fructose, and other monosaccharides or disaccharides present in fruit, okay, with different names. Type of sugar in the fruit, BQRST, with different mass, found in 50 grams of fruit. For B, 13.6 grams, found in 50 grams of fruit. For Q, 12.2 grams, found in 50 grams of fruit. So, the question now, calculate the percentage of the fruit that is made up of sugars. Show all your steps. Show all the steps in your working. Percentage with 3 marks. Firstly, in our workout, we have to sum all the masses of different sugars present in fruit by adding 30.6 plus 12.2 plus 3.4 plus 2 plus 0.4 equal 31.6. This is the first step. This total masses of sugar present in 55 grams of fruit. So, total fruit mass equal 55 gram of fruit. Okay? What about percentage of this sugar in relation to 50 gram? So we have to divide 31.6 divide 55 times 100 equal 57.5%. Okay? This is the percentage of sugars present in 55 gram of fruit. Double I. Draw a bar chart of the data shown in table 1.3 on the grid in figure 1.3. Each bar should be separated by each type of sugar, even if this second line not written in your exam. Please, when you draw bar chart, leave this space between column and column, bar and bar. Okay? Now, where is the table? Type of sugar in the fruit? This type called categoric table or graph. Why? Because here letters or types of sugar. Okay? So on x-axis we have to put type of sugar in the fruit. It's independent variable. Independent variables. Okay? What about mass of sugar in 55 fruit per gram on y-axis? It's dependent on the type of fruit of sugar. Okay? So now I divide x-axis into P, Q, R, S, and T. Okay? Now y-axis, the highest figure here 13.6 in y-axis. So I divide this y-axis into 2, 4, 6, 8, 10, 12, and 14 to cover the maximum figure here, 13.6. Now, how to plot bar chart? Look, for B, 13.6. Okay, 14.6. Yes, okay. No shading, no coloring needed in our exam, especially in graphs or drawing. No shading, no coloring. Okay, plain, but by your pencil and ruler. Now, leave space and take Q. For what Q? 12.2. For R, 3.4. For S, 2. For what? For T, 0.4. What about requirements when we draw bar chart? What's the precautions needed? Please, the width of each bar equal, like here and here, must be equal. Also, the distance left between bars separated by area of space equal also. Okay? This bar chart. Now, last question, III, name one source of sucrose in the plant and the process used for loading sucrose into the phloem tissue. The theoretical, from theory, from your study, like paper 2. Name one source of sucrose. May sucrose passes or translocated through phloem from what? From leaves. Growing, mature leaves act as source. Okay, leaves. Also tuber of potatoes, for example, tubers. Here we can write leaves. Okay, act as source of sucrose due to photosynthesis. And translocated through phloem tissue, sieve tube elements or cells. What's the process needed for loading? Active transport. Remind yourself, here, source like palisade cell, mesophyll cell, photosynthesis takes place, glucose, then sucrose is formed in sucrose. Okay? What happens? Companion cells pumps protons to the wall. Acidified wall, okay, by pumping protons towards the wall, it uses ATP. It uses energy from ATP. Okay? The energy due to hydrolysis of ATP needed for pumping protons to the wall. This helps protons, and protons increase in concentration in the wall and return back passively through co-transporter protein gate. But this co-transporter means protons attach with sucrose from palisade cells. Okay, so carry passively sucrose with it towards companion cells. Then at companion cells, the highest concentration of sucrose, okay, allow passage of sucrose from high concentration into low concentration in sieve tube element passively through plasmodesmata. Okay, thank you for watching. My best wishes for all of you. Good luck. Bye bye.